Hi, welcome to Science Hangout from Science Mission. Today we have Dr. Mike Pluth, Associate Professor in the Department of Chemistry and Biochemistry from the University of Oregon is with us. Dr. Pluth has won several awards. In 2016, he won University of Oregon Outstanding Award. In 2016, again, he won Community Reference Teacher Scholar Award. In 2015, Time Chemistry Journal Award. Again in 2015, Alfred Sloan Fellowship. In 2015, he won another award, NSF Career Award. And 2010, NIH Pathway to Independence Award. In addition, he also has won several awards during his postdoctoral tenure as well as a grad student. Welcome, Dr. Pluth.
Hi, thanks for having me.
So thanks for joining us.
Absolutely.
Can you please tell us about yourself, your career, your background, etc.?
Absolutely. So I'm a native Oregonian and was a chemistry and math major at the University of Oregon as an undergraduate student, and then went to graduate school at the University of California, Berkeley, where I really became interested in mechanistic chemistry and trying to understand how you can engineer molecules to carry out specific purposes that you want. And during that time, I became somewhat more interested in biological chemistry, and so moved to MIT as a postdoc, where I worked on developing chemical tools for looking at small molecules in different biological systems. And then I started my independent career in 2011 at the University of Oregon, where I've been developing chemical tools for looking at other small molecules in biology, mainly ones that evolve around sulfur.
So, you have a pretty good, very nice career. And can you please tell us about some of your mentors or the role models who supported you during your career?
Sure. So I've always been very fortunate to have very good mentors, even starting as an undergraduate, where I started as a researcher in the lab, having very supportive mentors who really fostered curiosity and made me want to think about big picture ideas and not just the small things I could do every day, but how these could relate to bigger questions. And then as a graduate student, I had the very interesting experience of having two PhD advisors. And the thing that I've found very interesting about that is they approached problems from very different perspectives, but almost always came to the same answer. And I found that really, really interesting and educational because it showed that there's not one right way to do things and not one right way to think about problems. And that really understanding the core issues involved really leads you kind of down the path of discovery. And so I think having people who have fostered creativity and been great mentors and great teachers throughout my educational career has been very important and very fun for me too, which I think is a big part of it.
So in addition to your, I know you are busy with your work and all, like research work. So do you do other things, other hobbies and interests other than research?
Yeah, so I really like photography and traveling, and those often go hand in hand. So I like to take pictures of different things. I also quite like gardening. So a lot of faculty friends have gardens as well, and we kind of compare notes and see who can grow the biggest, best tomato sometimes, I think. That's always a good distraction outside of the lab.
And let's talk about your lab and lab people. So can you please describe your lab and some of the postdocs, their students?
Yeah, so right now, my lab has one postdoctoral researcher, 3 undergraduates, and 7 undergraduates. And so we just had a number of graduate students graduate, which is great, and they have jobs, which is fantastic. And kind of the general standard state personnel count for my lab is probably 1 to 2 postdocs and 6 to 8 graduate students, and then about that same number of undergraduates. So for me, I think it's really important to have a mixture of researchers at those different career levels, because most people who are in graduate school or who are postdocs got their their start and inspiration in research as an undergraduate. So I think providing that opportunity for current undergraduates is very important to help foster kind of the next generation of scientists. And I think it's also good for graduate students to have experience in training younger scientists and postdocs, having the experience in training graduate students and undergraduates as well.
And how do you motivate them, like especially undergrads? Like, so
Yes.
like sometimes the research is kind of up and down. You don't get the right result every time.
That's right. That's right. So one thing that I tell almost every undergraduate who joins my lab is that in laboratory classes as an undergraduate, all of the experiments are engineered to work 95% of the time. And if you follow directions, you'll get the reaction to work almost all the time. Whereas in research, it turns out, in my experience at least, that about 95 of the reactions you run don't work. And so that's a very different experience. And you really have to kind of grow tenacious and understand that just because things don't give you the result that you were expecting or that don't work exactly how you thought they might, if you can learn something from that experiment, even if it's an experimental technique or a new way to think about data analysis, that was a successful experiment. And so I think that really trying to motivate students to expand their toolbox and develop a greater selection of tools that they can use to look at current and future problems is a good way to motivate them.
And how often do you interact with them?
How
like,
often
is
do
it
I
every day? Or?
Yeah. So I mean, I think most people in my lab would tell you the same thing, that you know, I often walk through the lab multiple times each day and stop and chat with people about the experiments that they're doing or new results that they might have, and you know, I'll come and find me in my office also. I think that one thing that's very interesting is in scientific training as an undergraduate, graduate student, and postdoc, you're in the lab doing the science, but then as a faculty member, when you are directing research, you aren't really doing much of the actual experiments anymore. And so it's fun to kind of live vicariously through the students sometimes in terms of experiments that they're doing in the lab.
And coming back to the research, what time point you got interested in hydrogen sulfide and its effect in?
Yeah, great question. So certainly, 10 years ago, if you told me that I was going to be interested in hydrogen sulfide and work with a lot of sulfur-containing molecules, I probably wouldn't have believed you. But when I was a postdoc at MIT, I worked with another small gaseous biologically important molecule, nitric oxide, or NO, and that really got me interested in trying to understand how these small reactive biomolecules play important roles in chemistry and in biology. And really that interface was very interesting to me. And when I was a postdoc, there was a lot of new literature coming out on hydrogen sulfide that it might be kind of the next important small molecule gas that's involved in chemical biology. And so as a graduate student, I had a lot of training in mechanistic chemistry and physical organic chemistry, which to me means trying to use mechanisms of chemical reactivity to get molecules to do things that you want them to do. And so I viewed that with my skill set, I could make an impact looking at this new important biomolecule by kind of carving out a niche at the interface of chemistry and biology, using some of these skills that I used and learned as a graduate student, but applying them in a new area of chemical biology.
So let's listen to your work on the sulfur-containing molecules.
All right, sounds good. Share the screen here. All right, can you see that?
Not yet. Yes, here it is.
Perfect, great. So what I'm going to talk about is really kind of some of the central roles of sulfur in biology and at the interface of chemistry and biology, which is what I am interested in. And so if you want to get an idea of recent things that come out from my group, we have a website down here that is updated frequently, and also a Twitter feed that comes out with new papers and things like that from our lab. And so if we think about sulfur, most of us have experienced sulfur before, right? So if we've ever been to a hot spring or a geothermal event or a volcano, you often have this kind of nice haze that has this somewhat sulfur odor. That's mainly a mixture of sulfur dioxide, so SO2, and small amounts of hydrogen sulfide, H2S. And if we look around here, we can see a few characteristics here. We see that there's a little bit of yellow color on the ground. That's actually elemental sulfur that's deposited on the ground. We also see that not a whole lot of vegetation is living around this hot spring, because it turns out that a lot of these sulfur molecules are very caustic and make it hard for different organisms to live there if they're in really high concentrations. And if we think about the roles that sulfur might have in biology, if we look at a periodic table and we think about elements that are involved in life, we often think about organic chemistry, which is the chemistry of carbon-containing molecules that have carbon, nitrogen, oxygen, and hydrogen. But we can really expand the elements that nature uses if we look at the bio-inorganic chemistry. So now including non-carbon-based molecules. We now have things like metals, so first row metals like manganese, iron, cobalt, copper, and zinc. These are often found in lots of different enzymes. We have other alkali metals like sodium, potassium, magnesium, calcium that are involved in signaling or in bones in the case of calcium. We also have phosphorus, sulfur, and selenium. And these are also important biomolecule constituents. But really what I'm going to focus on today is sulfur. And sulfur is actually the 7th most abundant element in the human body. And so it really plays a lot of important roles in biology. And if we look at how sulfur is involved in kind of the global cycle, it's really involved in a lot of different things. And so there's a lot of sulfur in the Earth's crust, for example. It's in a lot of different ores that we mine or in fossil fuels. Humans convert a lot of that sulfur in the ground into sulfur dioxide through combustion or through different industry processes. And then in the atmosphere, you get sulfates. These actually contribute to acid rain, which isn't always the best thing. This can get into the water. So we have things like sulfates in the water. And there are microbes in the water that will convert sulfate into different forms of sulfur that make it into the sediment that find other microorganisms. And then this can be taken up by plants. So there really is a global sulfur cycle that has a lot of different forms of sulfur in it. And in these, we can see one place where H2S is abundant, and that's actually from volcanoes. And it turns out that H2S and other sulfide-containing species are commonly found near hot springs, volcanoes, and places like that. If we look at common sulfur-containing biomolecules, one of the amino acids, cysteine, has a SH group on it. This is a very important amino acid. It's involved in lots of enzymes because we can have this SH group function as a very good nucleophile. So if there's an enzyme that carries out a reaction, often that may contain a cysteine. Also, this can be oxidized to form a disulfide. So these disulfides are very important for enzyme structure or in hair. It gives it some of the curliness in hair. Also have things like homocysteine or glutathione, which is a small tripeptide. This is the primary thiol in a lot of living systems, and it's really important in the redox balance. So if we think about an antioxidant, glutathione is nature's most abundant thiol-containing antioxidant. And then we also have things like methionine, which is an essential amino acid, meaning that we have to eat this, we can't make it ourselves. And this is also found in S-adenosyl methionine, which is an important enzyme cofactor for lots of different reactions. And one thing that I really think makes sulfur special and makes, or at least contributes to why it's so important in biology, is that it's really complicated. And we may think of that as a bad thing, but the fact that it's complicated means that it can actually have a lot of different oxidation states, meaning that it can either have or be missing a lot of electrons. And that is really important because a lot of reactions in biology involve electron transfer. So transporting electrons from one molecule to another, maybe from an enzyme to a substrate. It's the fact that sulfur can access so many different oxidation states makes it very versatile. And if we think about hydrogen sulfide, H2S, this is actually a weak acid, so it exists both as H2S and as an anion, a monoanion hydrosulfide. We also have a lot of different sulfur-containing species in biology. We saw thiols before. This could be cysteine. We can have sulfenic acids, which are an oxidized form of cysteine that are important in signaling. We also have nitrosothiols. These are important in the crosstalk between thiols and nitric oxide, another important small gaseous molecule. We have other oxidized forms of sulfur, like a persulfide, the strange SSH functional group. We have polysulfides. These are chains of sulfur atoms linked together. These can range anywhere from 2 or 3 sulfurs to much longer chains. We have other oxidized forms of sulfur like sulfoxides, elemental sulfur, or species with sulfur and oxygen in them. And there are bacteria that can essentially convert sulfur all the way from its reduced state to its fully oxidized state and back the other direction, which essentially gives a lot of organisms the ability to create or use energy. And as I mentioned before, much of my lab is interested in hydrogen sulfides. Let's talk a little bit about its basic properties. If you look for images of hydrogen sulfide, you'll often find a lot of these caution H2S signs. It turns out that in high concentrations, hydrogen sulfide is quite toxic. I'm almost sure that everyone has smelled hydrogen sulfide at some point in their lives. It's the classic rotten egg smell. So if you smelled that before, it's hydrogen sulfide. In water, it's a weak acid, which makes it actually which is a very important property because H2S itself can be deprotonated to form hydrosulfide anion, which is very, very water soluble. But this more hydrophobic H2S form can easily cross cell membranes and is very important in different biological systems. And it turns out that if we look at the chemistry of hydrogen sulfide, a lot of the chemistry has been well developed in geochemistry for a long time because a lot of very colorful minerals are metal sulfide salts. So cinnabar is a red mineral, it's mercury sulfide. Fool's gold or pyrite is iron sulfide. Zinc sulfide is this very nice blue color. So it's really a lot of geochemistry that's involved in sulfide as well. But I've mentioned that H2S and hydrogen sulfide are very important in life. And if we look at the history of evolution on Earth, we might get some clues as to why that is. And so if we look at the levels of hydrogen sulfide shown in green here throughout history, about 3.8 billion years ago, where the first microfossils were found, at that point there were medium levels of hydrogen sulfide, really no oxygen left, but a lot of iron. And then there was a great oxidation event, which is thought to be the formation of oxygen by cyanobacteria. And what happened here is we now have a lot of oxygen that is being formed. And this can interact with the iron and the sulfur that's around and actually increase the amount of free hydrogen sulfide in the oceans. And so during this time, organisms were evolving in the presence of really large concentrations of hydrogen sulfide. And then as oxygen concentrations go up, the O2 turns out to oxidize a lot of this hydrogen sulfide back down to sulfate and polysulfides, and modern animals are formed. But really for about almost a billion years, there was a lot of interplay between evolution, iron, and hydrogen sulfide. And this formed a lot of really important iron-sulfur centers, which are still found in almost all living organisms today and are a great way to transport electrons in different systems. And if we think back to the toxicity of hydrogen sulfide, it's just kind of a basic chart showing some of the toxicity. It's actually one of the compounds that the human nose is the most sensitive to. So we can detect hydrogen sulfide and identify it at about 500 parts per billion. And this is probably an evolutionary mechanism because H2S is often given off from bacteria that live in anaerobic or oxygen deficient environments. So maybe if you had a food supply that was not very safe, it might have hydrogen sulfide. So it makes sense that we can detect this at quite low levels. As you get up into higher concentrations, so parts per million levels, you get eye irritation, you start to get tissue damage, and if you get over about 1,000 parts per million, that can result in death. But at much lower concentrations, hydrogen sulfide is actually a very important biomolecule and joins other gases, nitric oxide and carbon monoxide, as what are called Gayssot transmitters. And so these are molecules that are small gases that are produced by an organism and serve a physiological function within the same organism. And so of these three molecules, hydrogen sulfide is certainly the most recent to be added to this group. And that's kind of why we became interested in H2S, because it's an area where I think chemists, biologists, and physiologists can really look at important problems and look at them from different perspectives. And so looking at kind of a history of H2S here, it's thought that about 250 million years ago, a large acidification in oceans resulted in release of H2S from much of the water on Earth, killing about 95% of terrestrial life. One of the first examples that I could find relating to H2S in the medical profession was in 1713 or the early 1700s, when it was shown that in London, sewer workers who complained of eye irritation often had black coins in their pockets. And at the time, coins were made of copper. We now know that's copper sulfide, which is a very dark color. And so it makes sense that workers who are exposed to high levels of chronic hydrogen sulfide had some physiological impacts of that. And then moving forward quite a bit here, what really got me interested in hydrogen sulfide was when I was a graduate student. I wasn't actually working on chemistry related to H2S, but there was a very interesting paper that showed that if you took an animal like a mouse and exposed it to about 80 to 100 parts per million of hydrogen sulfide in the absence of oxygen, that the mouse didn't die, but rather the mouse would go into a suspended animation-like state where its pulse and breathing would slow down quite a bit, and its core temperature would also drop. And you could keep the mouse in that state for a number of hours, remove all the hydrogen sulfide, put oxygen back in, and the mouse would wake up and not have any long-term damage. So I think that convinced a lot of people that H2S is really an important biomolecule. It's doing a lot of interesting chemistry in living systems that is worth figuring out. And so if we think about where hydrogen sulfide comes from, this may look fairly complicated, but really the key point here is that there are a number of enzymes that can transfer the thiol, or SH group, from things like homocysteine, which we saw earlier, or cysteine, and make hydrogen sulfide. And these enzymes are distributed widely throughout the body. Their distribution is somewhat tissue specific, meaning that there may be different ways to activate these different types of enzymes in different systems. And if we think about some of the roles of hydrogen sulfide in the human body, it's really quite important in a lot of different organ systems. This includes the cardiovascular system, the lungs, the brain, and the GI tract. And really almost every day there are new papers coming out showing new roles of hydrogen sulfide in different organ systems. And a few examples that I think are kind of interesting. So in one of an early example of a mouse model that is missing one of the key H2S producing enzymes, that means if it's missing one of these enzymes, it does not produce as much hydrogen sulfide as you would normally expect. This has shown that the tissues in the heart of the mice that are missing this key enzyme do produce less hydrogen sulfide than in a normal mouse. That makes sense. Also, things that are quite interesting here is that the blood pressure is somewhat modulated by hydrogen sulfide. And so as the hydrogen sulfide is increased, if you add hydrogen sulfide to these mice, it results in a decrease in the blood pressure of the mice. And the mice that are missing this key H2S producing enzyme are much more sensitive to this, suggesting that H2S plays an important role in blood pressure mediation. And then looking at the resting blood pressure of mice, we can see that the mice missing key H2S producing enzymes have a much higher blood pressure than normal mice, again, consistent with roles of hydrogen sulfide in blood pressure mediation. A few other examples that I think nicely highlight the diverse roles that hydrogen sulfide can play. This here is in wound healing. So if we look at the ability of mice to heal wounds, these are again comparing the animals that are missing one of the key H2S producing enzymes in black here versus the white squares are the normal mice. And this here is the wound size. And if we look at a normal mouse, it heals a wound at a certain rate. And these H2S deficient mice heal the wound much more slowly, suggesting that H2S may play important roles in wound healing. H2S is also very important in the gastrointestinal system. And one type of common drugs that are used are NSAIDs, so non-steroidal anti-inflammatory drugs. These are common over-the-counter painkillers, but these actually can do a lot of GI damage. And in a model here showing the gastric damage score, we see an increasing amount of gastric damage as we increase the amount of an NSAID that a mouse is given. However, if the NSAID now has an H2S releasing molecule appended to it, we see a much decreased damage score, suggesting that H2S may play beneficial roles in protecting the GI tract from damage. And then one final example here is in a model of kidney transplant. And one huge problem in the US and around the world is the viability of organs for transplant, and often transporting them causes organs to no longer be viable for a recipient. And so in this model, this is a kidney transplant model in mice. A kidney is taken out from the mouse and then stored for a while and put back in the same mouse. And we can see that not a lot of mice survive this. So after 5 days, almost all of the mice have died. Whereas if the organs are treated with hydrogen sulfide, we see a large increase in survival for the mice. And so I think that these three examples really highlight some of the diverse roles that hydrogen sulfide can play in different pharmacologies and possibly as therapeutic tools. So what my lab is interested in is really using chemistry as a tool to investigate hydrogen sulfide in biology. And so things that we are interested in, we're interested in developing new chemical tools for detecting, quantifying, and imaging hydrogen sulfide in different biological contexts. And so if you want to see where H2S is being produced, you need some way to image that or detect it. We're also interested in developing compounds that can be programmed to release hydrogen sulfide on demand. So give us a way to deliver H2S to a certain part of an organism. And then if hydrogen sulfide is important in biology, it must be doing some chemistry somewhere. And so we work on model compounds to try to understand the chemistry that's going on with hydrogen sulfide. So just tell you a few snippets of things that we've been working on in my lab, and the first of which will focus on developing chemical tools for detecting or imaging H2S in different biological environments. And one of the common tools that scientists use to look at molecules in live systems is fluorescence microscopy. And so these are molecules that can be engineered to release light in the presence of an analyte that you are looking for. And so this is a very common technique. And so we've been trying to develop chemical tools that can image H2S by fluorescence microscopy. And so ideal properties, you want imaging agents that are water soluble, non-toxic, selective for H2S over other things that might be around. So I often describe this as you're trying to pick out one specific type of molecule in a waste bottle. So there are many types of reactive molecules around, but you're only looking for one. We also want molecules that become fluorescent, so are initially dark and then become emissive when they find the analyte that you're looking for. So this is an illustration showing why we want a turn-on fluorescent probe. So in the top here, if we had all the probe turned on or emitting light, I've actually extinguished a lot of the lights in this picture, but you can't see where they are because there's so many lights being shown. Whereas down here against a darker background, it's very clear to see that these lights have been turned on, which if this were a fluorescent probe would tell you that H2S is being produced in this environment. And so we are chemists by training, and so really we tried to use the innate chemical properties of H2S to develop different tools for imaging hydrogen sulfide. And so really how we've done this is by making small molecules that have a reactive group that can react selectively with hydrogen sulfide to undergo a chemical reaction. And this chemical reaction can be translated into light. And so what this is showing here is that upon addition of hydrogen sulfide, this is a measurement of the light output or fluorescence of this probe. And as we add hydrogen sulfide, we see an increase in fluorescence, meaning that this compound is becoming fluorescent when it reacts with hydrogen sulfide. And we've developed a lot of different types of fluorescent probes for hydrogen sulfide detection. We've made compounds that are chemiluminescent. And one benefit of these types of molecules is that they do not require excitation or a light source to emit light, but they just glow on their own. So these can find hydrogen sulfide and essentially glow if H2S is around. We've developed very, very bright probes for hydrogen sulfide that can be used in zebrafish to look at H2S production, which I'll talk about here in just a minute. We've also developed very versatile ways to attach these sensors to different parts of a cell. So if you wanted to look at what was happening in one subcellular location, you could use this technology to deliver these probes to one part of a cell. We've also done studies to understand how these sensors work. So if we're going to use outcomes of this sensor technology to understand biological outcomes, we probably want to know how they work. So my group is a group of organic chemists, so we make molecules. The exact ways in which we make them are not completely important for this context, but we really take ideas and reactions and try to engineer them into developing molecules that we want to use. So all this is showing is here we can make this sensor. This is based on a very bright chromophore that can emit light. If this azide group or N3 group is here, this molecule does not fluoresce. Once it reacts with hydrogen sulfide, it becomes fluorescent. We can use this as a reporter for hydrogen sulfide. But things we need to worry about are really, there are a lot of other thiol-containing or SH-containing species or reactive species in general in a biological sample. So we've talked about things like cysteine or homocysteine or glutathione, other reactive small molecules as well. So we really need to make sure that these probes we develop are selective for the one analyte, hydrogen sulfide, that we're interested in, not other molecules. And so what we do commonly is we will test our fluorescent probes against hydrogen sulfide, then also against other reactive sulfur, oxygen, and nitrogen species. And this here is what we want to see. We have a large fluorescent turn-on for hydrogen sulfide, but really no turn-on for other reactive species. And so once we have probes that work very well, we'd like to increase their applications and try to use them not just in a cuvette or a test tube, but actually in a biological context. So as an example of this, we collaborated with Raghu Parsarathi's lab here at the University of Oregon. He's a biophysicist. And what they are interested in looking at is using 3D imaging techniques to look at the gut or the GI tract of larval zebrafish. To give you an idea of scale, this is a 5-day post-fertilization zebrafish. It's about 3 millimeters in length. And so what they can do is introduce a payload, which can contain anything you want, in our case, the H2S sensor, into the gut of this zebrafish. And then if we add this molecule, which I'm guessing almost everyone in the audience has actually consumed at some point, this molecule is diallyl trisulfide. It is a main sulfur component in garlic. It reacts with thiols like glutathione to give off hydrogen sulfide. So if we introduce the sensor and this H2S donor into the gut of zebrafish, you can see a large increase in fluorescence. We're looking at the fluorescence in the little box area here in this zebrafish, showing that we can detect the H2S released from these compounds inside the gut of the zebrafish. And so I mentioned that this is a common sulfur-containing molecule that is common in many things that we eat. And this has a polysulfide in it. Here's a trisulfide with 3 sulfur molecules. And nature actually uses this similar motif in lots of different molecules. And so we saw this molecule before, diallotrisulfide, found in garlic and onions. Some of these polysulfides are found in things like asparagus and mushrooms, and they're really much more exotic polysulfides as well. So the key thing I'm showing here is really there's a large diversity in sulfur-containing molecules in a lot of different natural products that can be isolated from different organisms. And so that's kind of highlighted here. Common things like mushrooms, onions, garlic, cacti, cruciferous plants often have these sulfur-containing species in them, which all release hydrogen sulfide. And so in terms of releasing hydrogen sulfide, we've actually been interested in developing new classes of hydrogen sulfide donors. And so again, this slide, the structures are not important, but just showing that there's a large diversity of structures that have been developed for releasing hydrogen sulfide. And key themes here is a lot of these H2S donor molecules have either polysulfides or other sulfur-containing species that can be activated to release hydrogen sulfide. So really what we've been interested in is developing new ways to be able to trigger the release of hydrogen sulfide on demand. And to do that, we've actually started looking at another sulfur-containing molecule, carbonyl sulfide. So we've all probably heard of carbon dioxide. This is like carbon dioxide, but one oxygen has been replaced with a sulfur. And carbonyl sulfide is actually the most prevalent sulfur-containing gas in the atmosphere. It's also been implicated in origin of life type chemistry because carbonyl sulfide is produced by volcanoes and hot springs and has been shown to actually facilitate coupling peptide bonds. You have an amino acid and have carbonyl sulfide nearby that can facilitate forming a new peptide bond. But why we're interested in carbonyl sulfide is there's an enzyme called carbonic anhydrase, which is expressed in essentially every living organism. Its main purpose is to detoxify CO2 in the body. And it does that so well and is so efficient, it doesn't care if you replace one oxygen on CO2 with a sulfur, and it will convert carbonyl sulfide into hydrogen sulfide. And so we've developed new ways to harness molecules that can release carbonyl sulfide as a new way to deliver H2S on demand. And the way that we did this was really trying to re-engineer well-developed chemistry that's been developed in the medicinal chemistry field as a way to deliver drug molecules. And so how this works is that here, labeled as FL, is a payload that we might want to deliver. And there's essentially a large protecting group that masks this payload. And so people have figured out how to activate this protecting group to undergo a cascade reaction, decompose, and release this payload that you might want, and also give off byproducts, which typically people don't really care about. But our thought was, well, why don't we change one of these oxygens into a sulfur? And if we did that, this would no longer release carbon dioxide, or CO2, but would rather release carbonyl sulfide, or COS. And so the design here was that, first off, we thought we could use this in sensing, where we could have some sort of molecule that could react with hydrogen sulfide, be activated, deliver the payload, and regenerate H2S, first by forming carbonyl sulfide. But really, this technology is very versatile because it allows us a way to engineer new ways to release this carbonyl sulfide, which then gets converted into H2S on demand. And as a few quick examples of this, we actually took carbonyl sulfide and treated carbonic anhydrase, this enzyme directly with carbonyl sulfide, we can detect H2S being produced. These enzymes are quite efficient in making H2S. We can also inhibit the enzyme with an inhibitor and reduce the rate of H2S production. And this doesn't just work in a test tube, it also works in live cells. So what we've done here is incubated cells with one of our H2S responsive fluorescent probes, and then introduced carbonyl sulfide. And the fluorescence increases in the cells, showing that the carbonic anhydrase in the cells is sufficient to generate H2S and activate the probe. So as one quick example of how this can be useful, one of the primary roles of hydrogen sulfide is thought to be protection against oxidative stress. And so what we did is we developed donor molecules that have a caged molecule of carbonyl sulfide, which will convert into hydrogen sulfide after activation, but also a triggering group that is stable until it finds reactive oxygen species. So we know that free radicals are often damaging. So this would find free radicals, be activated, and release H2S, which could then provide protection against those free radical species. And as a proof of concept, this works quite well. So we can treat these donors with hydrogen peroxides, let's say reactive oxygen species, and see an increase in H2S being released as we increase the amount of peroxide being added. We can also look in more detail at what reactive species are generating the H2S from these donors. And it's mainly hydrogen peroxide, superoxide, and peroxynitrite, all of which are actually commonly involved in immune response. So when immune cells think they're under attack, they often pump out large amounts of reactive oxygen species as a way of fighting that invading pathogen. And as an example of converting this basic chemistry from a test tube into live cells, we can take cells here. This blue stain is just showing where the nucleus is of each cell and shows that they're alive. If we incubate these cells with an H2S reporter that we developed in our labs, this molecule will be not fluorescent in the absence of H2S, but become fluorescent when it finds H2S. And also this reactive oxygen species donor. So in the absence of reactive oxygen species, we don't activate the donor, so we don't make any H2S. We don't see any fluorescence from our probe. If we add hydrogen peroxide, however, that activates the donor to release hydrogen sulfide, which is detected by our probe, and we see a large increase in fluorescence. And so this is in a system where we add hydrogen peroxide. We can also use a different cell line, so these are immune cells, and we can stimulate these cells to think that they're under immune attack and release reactive oxygen species. So prior to such activation, we don't see any hydrogen sulfide. But once we stimulate these cells to produce reactive oxygen species, that activates the probe, and we detect that with our sensor. And why we're really excited about this is we can actually impart protection on cells against oxidative stress. And so what this graph is showing is cell viability, so how many cells are surviving, as we increase the amount of hydrogen peroxide being added to these cells. And we know that hydrogen peroxide is a reactive oxygen species. It's quite toxic. And so we see a decrease in the amount of viable cells. At about 100 micromolar, we only have about 30% of the cells still alive. However, if we incubate these cells now with our donor, that will be activated by the peroxide and deliver H2S, we see a dose-dependent increase in cell viability, showing that these donors are able to protect the cells against the oxidative stress that the peroxide introduces. And then using control compounds, we can show that we don't see a large increase in cell viability if we have a molecule that lacks the sulfur or that lacks the reactive oxygen species triggering group. So we can confirm that this cellular protection is due to the release of carbonyl sulfide and hydrogen sulfide from these donor molecules. And it was kind of neat because this paper actually got quite a bit of publicity outside of the scientific community. And so this was picked up by a number of news sources, essentially with the idea that we've developed a new type of scaffold that can sense oxidative stress, so things that are usually cytotoxic or damaging, and then provide protection for those cells. And so we're trying to expand this from basic cell culture models into more complicated cell culture models and also into animal studies. So as an overview of what we do in my lab, we do a lot of different types of chemistry. So we're really an interdisciplinary group. We do some analytical chemistry. So I talked about that with our detection method, so making fluorescent probes for hydrogen sulfide detection. We also are interested in bio-inorganic chemistry, so understanding the basic chemistry that's occurring when hydrogen sulfide is being released, and also chemical biology, so developing donor molecules that can influence and impact the innate biology that's occurring when H2S is around. And really, all of these things combine to inform in context. And so our goal is to take chemistry that occurs in a test tube, engineer that to have an important biological purpose, and then use that in cell culture and model organisms to really understand what hydrogen sulfide is doing and hopefully make tools that can have some sort of long-term pharmacological potential. And I've really had a fantastic group of students in my lab who do all the work. So we have a combination of graduate students and postdocs and undergraduates. We've been very fortunate to have funding from a number of agencies, including the NIH, the NSF, and then also private organizations as well. And so again, you know, I'd really like to thank you for the opportunity to share what we do in my lab, and we'd be happy to answer any sort of questions you might have on things we work on.
Thank you, Dr. Pluth. So can we just get away from the slides?
Yes.
Okay, thank you. It's really, really interesting talk. Really, like I have worked on this hydrogen sulfide and all those things, antioxidants, but like learning more about what are the things which go into hydrogen sulfide, what would happen, and all those. I have some few questions about some of the things.
Yeah.
The first question would be the first slide, I guess. Like you showed that fumes of hydrogen sulfide and other sulfur-containing things, vegetation around that. But if you see the front side, there is some vegetation. Does it mean that that doesn't move, or it is not much diffusible? How does it go?
Yeah. So great question. So the question is essentially how do things live near these high sulfide environments? And so I think kind of there are two answers there. One is that you didn't see a lot of vegetation real close in where these sulfur gases were being released. Also, if people are allowed to walk that close to that environment, like I was to take that picture, there really are not very high levels of these sulfur-containing molecules being released from these springs. And so it turns out that the human nose is very, very sensitive to these molecules, and so we can detect them at much lower levels before they're toxic. But also organisms that live in those environments are well adapted to withstand higher sulfur contents. So for example, in mangrove swamps, so these are kind of the murky waters near mangroves, a lot of the clams and other organisms that live in that environment that often has a really high sulfide level, things that would be quite toxic to us, those organisms have developed defense mechanisms or evolved so that their enzymes are not deactivated by this high level of sulfide. So certainly that there are organisms that can live in those environments, but they're often very adaptive.
And one of the other question would be like, you showed some of the work in the cell lines. So using these hydrogen sulfide donors and all. So have they been used in the animal models or anything like that?
Yeah, so we haven't done much animal work from our lab. All the animal work that we've done has been through collaboration, but certainly there have been many studies out there showing the potentially beneficial effects of hydrogen sulfide or hydrogen sulfide donors, especially in situations where there's ischemia, so loss of blood flow to an organ or limb or tissue. And it's thought that hydrogen sulfide can really provide protection against a lot of these reactive oxygen species that proliferate in these types of environments. And certainly there's a lot of nice basic research and applied research going on in these contexts to really understand what sulfide and other sulfur-containing molecules are doing and how that can be leveraged to hopefully develop compounds that have some sort of therapeutic use.
And one more aspect of this sulfide is like you had mentioned about onions, garlics, and all. They contain multiple sulfide molecules and all those things. So, like we eat like humans eat a lot of onions, garlic, and all. Like, and I've heard like anecdotally that okay, there are some protective and all those things, but nobody has shown what happens inside the body.
Yeah. So
basically, yeah.
Go ahead.
Yeah, question would be like, are these sulfide molecules are broken down? Do our system have these enzymes that can break down some of these multi-sulfide molecules and use it as a beneficial effect for whatever development?
Sure. So certainly people have shown that, for example, garlic, if you consume garlic, or in mouse models, if the mouse consumes garlic, that does increase the free sulfide circulating levels in blood. And so certainly there is a way for that polysulfide to deliver its H2S and other sulfur species into the organism. Certainly some of these precursors are going to be more reactive and probably will get metabolized quite quickly. But it is a very bioavailable source of sulfide, which I think is quite interesting. And one thing that I've found quite fascinating is that a lot of the kind of traditional medicinal uses of a lot of plants that contain polysulfide species, what those plants were used for before modern medicine is often things that we now see a beneficial effect from H2S against. So for example, whether it be reducing inflammation or things like that. So I think that suggests that there probably is more to the story here, and that a lot of these donor compounds or plants that contain these high levels of polysulfides may have a way to deliver H2S to people when they eat them.
So we don't know still if any enzymes are there that can metabolize these polysulfides in the body or not. Do we know?
I don't know. So I mean, I know that the amount of sulfide that is released and metabolized from eating these things does increase the amount of sulfide. But I think that there still is a lot that we can learn about those systems.
Okay, I think there is a lot of interesting stuff needs to be done still. So
Absolutely.
this is really interesting work, and I really appreciate you taking time and be on Science Hangout and show your work. Thank you so much.
Yeah, thank you for the opportunity.
Look forward to more of your great work in the coming weeks and months and years.
Great. All right.
Thank you so much.
Great. Thank you.